So before we proceed to solving the assignment, we'll first see all the topics that were covered in week 5. So lectures 13, 14 and 15 were covered in this week, and lecture 13 was about aggregation of proteins and their effect, characterization of protein aggregation, factors affecting the aggregation, intermolecular forces for aggregation. So basically in this lecture we understand aggregation of proteins. The proteins are usually in their native state where they have which is stable and they have their actual functioning in its native state. And this once this native 3D structure is disrupted due to some factors, maybe internal or external, it leads to aggregation of proteins. So the protein goes from its native state to unfolded, starts unfolding due to the external factors, and then it leads to oligomers, and then it will lead to aggregation, which are insoluble aggregates of these proteins. So when there is a change in the conformation of the native structure of the protein, it slowly leads to aggregation of the protein. Next, we learn about the effect of this aggregation. So once proteins have aggregated, actually once they have unfolded, they start losing their function. Function is only there when it is in its native state. Once it gets unfolded and gets to forms aggregates, it loses its function. So that is one of the challenges of this aggregation or effects. And sometimes these protein aggregates leads to diseases such as Alzheimer's, prions, etc. So so it is really important to understand uh, and characterize this protein aggregation so that it can help us treating these diseases, or in understanding the effect of these aggregates in these diseases. And that we can do, that is discussed by characterization of protein aggregation in this topic. Several methods were discussed: thioflavin T fluorimetry, fluorescence assay, thioflavin T fluorescence assay, residual protein analysis, and then hydrodynamic diameter, morphological analysis. So in the lectures, in more details, these will be covered. But to give a summary, thioflavin T, here the thioflavin, it's a dye, it's a fluorescent dye that binds to beta sheet structures that are more prominent or they are more expressed in the protein aggregates. So as the protein gets transformed from native to aggregates, you can see these thioflavin dyes will bind to more of these aggregate structures than the native structures, and after binding they give a fluorescence emission. So in this assay we can monitor even the unfolding to aggregate formation of these native proteins by measuring the fluorescence. And then comes the uh, residual protein analysis. In this method we analyze the remaining protein that is left by simply using the protein quantitation assays such as BCA, Bradford, or Lowry method. And then the next method is hydrodynamic diameter. So there is a change in hydrodynamic diameter as well, which we can determine using DLS or transmission electron microscopy. And then there is also morphological analysis. In morphological analysis, again fibrils are formed, and fibrils are formed by observing this fibril formation. We can see. Then there is one more analysis of structure, analysis of change in the structure. So the native proteins have a defined 3D structure which changes upon unfolding and aggregation, and this can be determined using techniques such as FTIR or circular dichroism technique. So all of these techniques were discussed in more detail in the lectures. And then also the intermolecular forces factors affecting this aggregation was discussed. So there are several factors that can affect protein aggregation: pH, temperature, ionic strength, its solubility, shear, and adsorption surface. Some cases were also discussed for these parameters and how they affect the protein aggregation. But just to give a idea, so pH, so these proteins have specific residues. Each protein has a R group. The amino acids of the protein have a R group. These R groups can be positively charged, negatively charged, etc. And depending on the changes in the pH, they can gain more ions or lose ions. That will change the overall charge on the surface of the protein, and it will affect or might lead to protein aggregation. Similarly, temperature, uh, ionic strength, shear, solubility, adsorption surface also affect the aggregation of the protein. Next, also the intermolecular forces for aggregation was discussed. Van der Waals force, hydrogen bonding. All of these forces also affect proteins. When the proteins are exposed to a solution, they are covered by charges of ions, positive or negative, and they form electrical double layer and this stern layer, and these equation for stern layer and these things were also discussed for the in under the topic intermolecular forces for protein aggregation. And in lecture 14 it talks about how to determine the rate of aggregation, how to monitor the aggregation kinetics, ways to understand the aggregation in bulk and presence of surfaces, and experimentally how to understand the kinetics, all these things. So basically, the protein aggregation follows different pathways, and if this is the native state, it will first undergo intermediate, then unfolding, and or from native state it can also go to a chemically degraded form, or from unfolded as well it can go to a chemically degraded form. Then these intermediate forms aggregate. Similarly, the degradation products as well will form aggregates, and this will then lead to formation of precipitate. So these are the common protein aggregation pathways mostly protein follows. And then if we look at the protein aggregation in bulk, so in bulk, the due to external factors, as we discussed in the previous lecture about pH, temperature, solubility, shear, all those things. So all those external factors all those external factors mainly lead to conformational change in the protein structure. Conformational change. And then these conformational change will lead to formation of nuclei. Formation of nuclei, and finally it will lead to formation of aggregates. So all these steps, that is action of the external factors followed by conformational change and nuclei formation, all of these happens in the lag phase. So basically this is the lag time, and what we see is the only the final product, that is the aggregation of the protein. Then we discuss how these protein aggregation, what kinetics it follows. So it basically follows a second order kinetics. Second order kinetics, that is these molecules combine together, form bigger aggregates, that can be given by this equation, minus dn by dt equals to k n square, where k is the rate of aggregation. So if we further integrate this equation, we'll get a simplified form, that is at any time t we'll have n naught by 1 plus n naught kt. This expression describes the rate of aggregation in bulk. Also, we see that the rate of aggregation K can also be related as 8 by dA, where d is a diffusion coefficient, a is the radius of the sphere of the aggregates, and using the Stokes-Einstein equation, that is the diffusion coefficient d equals to kbt by 6 by eta A, where eta is the viscosity. So by relating these two equations, we get that the kinetics or the rate of aggregation is inversely proportional to viscosity of the solution. Next in the lecture, aggregation process on the surface was discussed. So whether the surface is hydrophilic or hydrophobic, the rate of aggregation can differ. For example, the proteins, there is less protein absorption in case of hydrophilic surfaces, while there is high protein adsorption in case of hydrophobic surface. So these protein adsorption will also lead to aggregation of the proteins. So this is how the surface of the protein can affect the aggregation process as well. And then determination of rate aggregation. So as discussed in the previous lecture, the thioflavin T assay can be used because it binds to the beta sheet structures. There are characteristics of aggregated proteins. So using this dye and getting the fluorescence, we can determine the rate of aggregation as well. Also in this lecture, modeling of protein aggregation was discussed, wherein when a surface, or typically in this case you can say a bioimplant, when it's implanted in a body, it's exposed to water and different kind of protein solutions. So the initial arrangement of the protein on the surface will be different, and with time these arrangement changes and the protein arrange in such a way that maximum number of proteins are adsorbed on the surface. So the arrangement, there is reorientation of this protein arrangement on the surface. So these things were discussed in lecture 14. And then in lecture 15, the effect of surfaces on the aggregation of protein was discussed. So in this lecture, uh, a model protein was taken, that is insulin. So basically a specific case was discussed wherein insulin was selected as a model protein, and then how this protein how the surface properties such as hydrophilicity, roughness, etc. affects the protein absorption and aggregation process, the morphology, structure of the protein, and these were uh, from the experimental results, these were all found out. Then also the surface properties such as roughness and hydrophilicity, mainly these two properties were uh, discussed. So the in later part of the lectures, how the surface properties correlate with this roughness or hydrophilicity with respect to protein aggregation rate was discussed, and then different approaches to inhibit aggregation. And in this, the beginning, mostly the experimental uh, to study the changes or the effect of protein aggregation on surfaces experimentally, such as using the external factors, temperature, the protein was exposed to change in temperature, and then the aggregation process was studied. And the thioflavin T that we discussed in the earlier lectures was used to measure the rate of aggregation of protein. And using other techniques such as AFM, uh, the modified how so a series of modified surfaces were taken, 4 or 5 surfaces, and the aggregation of protein on these surfaces was studied, and the morphological analysis was done by using an atomic force microscope. So these were all the topics that were covered in lecture 13, 14 and 15, which was mostly based on aggregation kinetics, basically protein aggregation, aggregation kinetics and determination of aggregation and their effects on aggregation of protein. And then now we'll discuss about the assignment problems. So this was the first question. Which of the following sequence represents the event of protein aggregation? Did you answer this only? Native, unfolded, oligomer, and aggregate?
Yes.
Yeah. So this is the correct answer. For the usually the protein would be in native state, but due to some external factors, it will get converted into unfolded, and then it will form oligomers and it will become an aggregate. Now the next question was aggregate results in formation of the following secondary structure. So during the transformation of these proteins to from a naive state to misfolded or um, misfolded state, it involves transformation of the alpha helical structures to beta sheets. So the correct answer here would be beta sheets. These beta sheets then interact further and form aggregates. Which of the following diseases arise due to protein aggregation? So the options given were Alzheimer, Parkinson, prion, and all of these. Correct answer would be all of these. So the Parkinson and Alzheimer's are basically neurodegenerative diseases that occurs due to misfolding of proteins, and prion is another such disease wherein prion is basically a protein that can trigger normal proteins to in the brain to fold abnormally, and it can affect both humans and animals. And the most common form of the prion disease is Creutzfeldt-Jakob disease. So here the correct answer would be all of these. The next question was which of the following methods can be used for characterization of protein aggregation? And the options were thioflavin T fluorescence, morphological analysis, residual protein analysis, and all of this. Again, the correct answer would be all of these. So, thioflavin T can be used. This basically detects the beta sheets, and more beta sheets are the alpha helices are converted to beta sheets, and that leads to protein aggregation. So more the beta sheets, more protein aggregation. That is how it is related. So thioplavin D basically detects these beta sheets in the protein aggregates. And then morphological and residual, the residual protein analysis will basically show the remaining bulk concentration of the protein that decreases with time. And morphological analysis, you can do AFM or AFM like and microscopy. So uh, in microscopy, you'll basically get to know uh, these protein changes in the protein structures that consist of the morphological analysis. So here the correct answer would be all of this. Now Next question was, which of the following equation is applied in dynamic light scattering method? So here Stokes-Einstein equation would be the correct answer. So dynamic light scattering is basically a laser beam is passed through and the fluctuations of the scattered light are detected. And the Stokes-Einstein equation, that is dH equals to kBt by 3 pi eta d, where kb is Boltzmann constant, T is the absolute temperature in Kelvin, eta is the viscosity, and D is the diffusion coefficient. So this gives a diameter dH, and based on that, uh, we can also analyze the protein aggregation. And next, this was the question: which of the following, which of the intermolecular force is or are involved in intermolecular interactions? Again, all of these would be the correct answer. So the dispersion forces, that includes London and van der Waals forces. The electric double layer, that is when material is exposed to the body fluid and initial proteins get attracted to this material, and then one more round of proteins will form the second layer. So there is a electrical the solutes will the charges of the solutes will come and coat the material, so these charges would form electrical double layer. And the short range forces such as the hydrophobic bonding, hydrogen bonding, and ion pairing and dipole interactions are also involved in the intermolecular interactions. Now, this was the next question. What is the first step of protein aggregation on solid surfaces? So, here the correct answer would be adsorption. Uh, First, adsorption takes place, and then the next steps of unfolding and aggregate formation will take place. Sakshi, I hope you're still with me.
Yes, yes, I'm following.
Okay. So this was the next question. Which of the event occur during the lag phase of protein aggregation? So here the correct answer is conformational change. So after experiencing external factors, the native protein uh, undergoes conformational change. Then it forms nuclei and formation of aggregates happens. So the correct answer would be conformational change. And this is the next question. What is the effect of the initial surface roughness on protein adsorption? So here the correct answer would be increases. So the initial surface roughness increases the protein adsorption. Uh, this graph also shows wherein the experimental results where in one of the experiments, this is what it was observed, that the more the surface roughness, the more adsorbed mass was there. And also if you see it like this, the more the smooth surface, it will give less chance for the proteins to interact with the material than a slightly rougher surface. So more surface roughness will increase the protein adsorption. And this was the next question. Which of the followings will cause protein aggregation? The options were upregulation of chaperones, interference with post translational peptide changes, inhibition of aggregation. None of these. Uh, Sakshi, uh, what did you answer for this question?
I answered none of these, but it seems that
Right.
upregulation of fat. But then in the
Yeah,
video,
I wanted to say that you guys can put in the discussion forum saying that the answer would be none of these, because even I think it would be none of this, because upregulation of molecular chaperones will basically convert the unfolded into more folded proteins or native proteins, right? As it was taught in this slide.
so how do I have to put it in the disc? Like from where?
Uh, I'm not really sure, but I had I had I think there is a discussion forum where you can mail or put in that forum saying that the correct answer for this would be none of these. And you can also tag the lecture there in saying that in this lecture it was taught like this.
Okay, okay.
Okay. And I have some sample questions for week 6 assignment. So, for week 6, this is the first question. Which of these is the correct sequence for homeostasis? Can you answer, Sakshi? I'll give you some time to go through this question. So, the steps are formation of fibrin plaque, vascular spasm, activation of coagulation cascade, and then formation of platelet plaque. But what will be the correct sequence?
Fourth option.
Fourth option, formation of fibrin plaque, then activation of coagulation cascade. Hmm. Okay. No, the correct answer would be third option. So first, vascular spasm will be there near the tissue injury, and then that will be followed by formation of a platelet plug, and then eventually it will activate the coagulation cascade, and lastly there will be a formation of fibrin plug. So the correct answer would be the option 3. Okay, I have the slide as well. So yeah, vascular spasm will lead to constriction of the blood vessels, and then more and more that will lead all the platelets to the side, and the platelets will form a plug, and then that will trigger the coagulation cascade, and more clotting factors will form the fibrin clot, the final fibrin plug actually, and then eventually that will clog the That is how healing process happens. And this is the next question. Where do all pathways of complement activation converge? C4 and C5 convertase, C2 and C3 convertase, C3 and C5 convertase, or C2 and C4 convertase. Do you want to answer, Sakshi?
I don't know this.
Okay, so the correct answer here would be again option 3, C3 and C5 convertases. So, if you go through the lectures, here it shows the classical pathway, the lectin pathway, and the alternate pathway. All of them converge near the C3 and C5 convertases. And this is the next question. Which of the following is not true about integrins? The options are they are transmembrane receptors, they mediate attachment of cells, they are important in cell signaling, and they are part of the ECM.
Fourth one.
Yes, correct. So they are not part of the ECM, but they help in mediating attachment of the cells to the ECM, and they are transmembrane receptors, and they also help in cell signaling. So fourth is the correct answer. Okay, these were all the questions I had today. And if you have any doubt, we can go through that. Or else, if you want me to repeat any other question.
I don't have any doubts.
Okay. Then let's meet next week again with week 6 assignment, solved assignment.
Yes.
Okay.
Thank you.
Thank you for joining.